physiology, as it's often taught, especially in those early years of medical training, can feel very linear.
Yeah.
It's a series of equations.
Exactly. It's cause and effect.
Right. You have a stimulus, you get a response. Input A leads to output B. If your blood pressure drops, the baroreceptors fire, your heart rate goes up. It's all very logical, and it's designed for homeostasis.
It's designed to keep the line flat. That's the goal of most systems we study: renal, cardiac, respiratory, stability. They fight to keep the internal world exactly the same.
But today, we are diving into a system that just fundamentally rejects that model. We're looking at female reproductive physiology. And as I was reading our source material for this, specifically the chapters on reproductive development and function in both Ganong's and Guyton and Hall, I was just struck by a completely different design philosophy.
Oh, absolutely.
This is not a system written in straight lines. It's written in circles.
It is a story of rhythm. That's really the defining characteristic. If you try to understand the system as a static, stable thing, you will completely miss the point.
You will fail.
You will. It's a time-dependent, cyclical dialogue. It's this intricate conversation between the brain, the pituitary, the ovaries, and the uterus. And unlike, say, the heart, which beats second to second, this rhythm plays out over days and weeks and even decades.
And that's exactly what makes this topic so challenging, but I think also so beautiful to unpack.
Yeah.
We aren't just memorizing a list of hormones. We're trying to understand a logic gate that literally changes its own rules halfway through the process.
That is a perfect way to put it. The rules of feedback loops, which for most of the month are there to keep things quiet and stable, they suddenly flip. They become explosive.
To create this one singular event, ovulation.
Exactly.
So here's our mission for this deep dive. We're going to try and synthesize the two perspectives of our source texts. On one hand, we have Ganong, which for me feels like the wiring diagram. It gives us the cellular mechanisms, the receptor interactions, the two-cell theory.
It's the nitty-gritty, the molecular level.
Exactly. And then on the other hand, we have Guyton, which pulls the camera back and shows us the systems engineering. Guyton is just brilliant at explaining the flow of it all, the regulation, how these tiny cellular events add up to create a functional cycle that can actually prepare for and sustain a pregnancy.
It connects the dots.
So we're going to build a conceptual map. And a quick disclaimer here, physiology is dense. What we're doing is creating a scaffold. We're interpreting, we're synthesizing, so that when you go back to the text, and you really should go back to the text, you aren't just reading isolated facts.
Right. You're fitting them into a structure you already understand. Think of this as the architectural tour before you study the blueprints. I want to help you see the logic behind the whole design.
Okay, so here's the roadmap. We're going to start chronologically. Section 1, the blueprint, how the system is even built in the embryo.
From the very beginning.
Section 2, the awakening, how puberty turns the whole machine on. Section 3, the engine. That's the ovarian cycle and the frankly brutal competition between follicles. Then Section 4, the control room, which is all about those hormonal feedback loops we mentioned. Section 5 will be the responder, which is what the uterus is doing while the ovary is so busy. And finally, Section 6, adaptability. What happens when the cycle is interrupted by pregnancy or when it ends in menopause?
It's the full arc, from conception to cessation.
Let's start at the absolute beginning, embryology. There's a concept in Ganong that I find just philosophically fascinating, and that's the idea of the default state.
It's really the golden rule of sexual differentiation. The foundational principle is that in human embryology, the default trajectory of development is female.
Meaning if you do nothing.
If you leave the embryo alone, if there are no specific active instructions to do otherwise, it will build a female body.
Which is so interesting because genetically we think of it as a coin toss.
Right.
You get an X from mom, you get either an X or a Y from dad. It feels 50-50.
Genetically, it is. You have your 44 autosomes and then your sex chromosomes, either XX for female or XY for male. But genes are just the library. They aren't the construction crew.
Right. They're the plans, not the action.
Exactly. For the first few weeks of gestation, the embryo is what we call bipotential.
It hasn't committed yet. It could go either way.
Precisely. If you were to look at an embryo at about 5 or 6 weeks, you'd see a primitive gonad. It's not a testis, it's not an ovary, it's just a gonadal ridge. And this is the critical part, you see two completely distinct sets of internal plumbing sitting right next to each other.
So it's like it has two possible futures laid out inside of it.
It does. You have the Mullerian ducts, which are the precursors to the female internal organs, the uterus, the fallopian tubes, the upper part of the vagina. And right beside them, you have the Wolffian ducts, which are the precursors to the male internal organs, the epididymis, the vas deferens, and so on.
So at 6 weeks, every single one of us had both of those blueprints just sitting on the desk, ready to go.
We did, and this is where the Y chromosome finally asserts itself. The Y chromosome carries a very specific, very powerful gene called SRY.
The sex-determining region of the Y.
Yes, it sounds like a military command, and it basically acts like one.
Huh, it does.
The SRY gene produces a protein, a transcription factor, that binds to DNA and acts as the master switch. It's the first domino. It tells that primitive, indifferent gonad, you are going to become a testes.
And once you have a testes, the whole game changes. Because unlike the primitive gonad, the testes is an active, hormone-producing organ.
It is not passive. To build a male, that newly formed testes has to do two very specific jobs. It's like a foreman on a construction site shouting two different orders at the same time.
Okay, what are the orders?
Order number 1 is build the male house, and order number 2 is demolish the female house.
Let's break those down. Build the male house. I'm assuming that involves testosterone.
Absolutely. The Leydig cells in that fetal testis start pumping out testosterone. This testosterone acts locally to stimulate the Wolffian ducts, telling them to grow and differentiate. They become the male plumbing. Without that testosterone signal, the Wolffian ducts just wither and die.
Okay, so that's the construction part. What about the demolition? Because you just had testosterone, you'd have the male system, but the female system would still be sitting right there next to it.
You might have a mess. You'd have both. So the testis has a second cell type, the Sertoli cells, and they secrete a second hormone. It's called Mullerian inhibiting substance, or MIS.
Physiologists are wonderfully literal with their naming sometimes.
They really are. MIS does exactly what it says on the tin. It targets the Mullerian ducts, the potential uterus and fallopian tubes, and causes them to undergo apoptosis. Programs cell death. They just shrivel up and disappear.
So to be clear, male development is a dual active process. You have to actively push the Wolffian system forward, and you have to actively destroy the Mullerian system.
Push and pull, build and destroy. Correct.
Now let's flip it. Let's look at the female side. An XX embryo. No Y chromosome, so no SRY gene.
And with no SRY gene, no master switch gets flipped. No testis forms. That indifferent gonad just sort of slowly, passively differentiates into an ovary. But here's the key distinction that Ganong really emphasizes. The embryonic ovary is quiet.
It's not secreting some kind of feminizing hormone to build a uterus.
No. That's a common misconception. It's not yelling any orders at all. The female system forms because of the absence of the male orders.
So because there's no testosterone.
The Wolffian ducts, the male potential, they just starve and regress. They disappear.
And because there's no MIS?
The Mullerian ducts, the female potential, are allowed to survive. They aren't destroyed. So they spontaneously continue their development into the uterus, the tubes, and the upper vagina.
So the female internal structure is the result of the system just being left to its own devices. It's the path of least resistance.
It is the default state.
There's a clinical condition mentioned in both texts that, for me, just solidifies this concept perfectly. Androgen insensitivity syndrome, sometimes called testicular feminizing syndrome. It's like the ultimate proof of this entire mechanism.
It is the thought experiment come to life. It's a fascinating and tragic example of this cascade.
So walk us through it. What's going on?
In androgen insensitivity, you have an individual who is genetically XY. They have the Y chromosome, they have the SRY gene, so their gonads develop into testes, usually undescended in the abdomen.
Okay, so the first step happens correctly.
It does, and those tests are functioning perfectly. They are making plenty of testosterone and plenty of MIS. The signals are being sent. The foreman is yelling both orders.
Build the male house.
But the workers have headphones on. The genetic defect is in the androgen receptor. The cells of the body literally cannot detect the testosterone. It's floating around in the blood, but the receptor protein that's supposed to grab it is either missing or non-functional.
So from the body's perspective, it's as if there's no testosterone at all.
Exactly. So what happens? The Wolffian ducts, the male plumbing, they're waiting for that testosterone signal to grow.
They never hear it.
They never hear it, so they wither away and disappear. No internal male organs.
Okay.
Now, look at the external genitalia. The penis and scrotum also require a testosterone signal to form from the primitive structures. Without that signal, the external genitalia follow the default path.
The female path.
Right. So they form as a clitoris, labia, and a lower vagina. The baby is born externally, looking perfectly female.
But wait a minute, the testes were also making MIS.
They were, and MIS uses a totally different receptor, which is not defective, so the body can hear the MIS signal.
Oh, no.
Yes, so the MIS does its job perfectly and destroys the Müllerian ducts.
That is just tragic logic.
What?
The testosterone couldn't build the male parts, but the MIS successfully destroyed the internal female parts.
Correct. So you end up with an individual who looks female externally, identifies as female, but has a blind-ending vagina, no uterus, no fallopian tubes, and undescended testes in their abdomen.
And this often isn't discovered until they're, 16 or 17, and go to a doctor because they haven't started menstruating.
Exactly. Because there's no uterus to have a period from. It really just hammers home that genetics is only the blueprint, hormones are the contractors, and the receptors are the walkie-talkies. If the communication breaks down, the whole construction project fails.
It changes how you think about biological sex. It's not a coin flip. It's a cascade of checkpoints.
A developmental cascade.
So, okay, let's fast forward. Let's assume the construction goes according to plan. We have a female infant born with ovaries, a uterus. The whole system is in place. But for the first, say, 10 to 12 years of life, it sits there, dormant.
The sleeping machine.
Is it broken? Or is it just waiting for a signal?
It's not broken at all. And Genong is very clear on this. The hypothalamus, the pituitary, the ovaries, they're all capable of functioning in a newborn. If you are to experimentally inject a child with the right sequence of hormones, the ovaries would respond. The machinery is fully functional.
So it's not that the car has no gas.
It's that someone has pulled the emergency brake up as hard as it can go. The system is being actively suppressed.
How? What's holding the brake?
During childhood, there's what's called a gonadostat in the brain. It's a neural circuit that is incredibly sensitive to negative feedback. Even the tiniest, almost undetectable amounts of steroids present in a child's blood are enough to completely shut down the brain's release of GnRH.
GnRH being gonadotropin-releasing hormone, the master hormone from the hypothalamus.
The one that starts the whole cascade. So that plus direct neural inhibitory pathways keeps the entire system quiet. It's being held in check.
Until puberty. The awakening. This is our section 2. What finally releases that brake?
That is one of the great remaining mysteries of neurophysiology. We don't know the exact molecular trigger, the ultimate start signal, but we know the mechanism of what happens when the brake is released.
Okay, what's the mechanism?
The inhibition lifts. The hypothalamus, specifically a region called the arcuate nucleus, starts to secrete GnRH. And this is arguably the most important concept in all of reproductive physiology. It doesn't just turn on a faucet and let it flow continuously.
Right. This is where pulsatility comes in.
It's a non-negotiable. The hypothalamus contains a pulse generator. It's a neural clock that fires, releasing a burst of GnRH roughly once every 90 minutes or so. It's a rhythmic beat, a pulse.
Why is that so important? Why pulses? Why not just a steady, continuous stream of the hormone?
It's a brilliant example of receptor biology. If you expose the pituitary gland, which is the target of GnRH, to continuous, steady levels of the hormone, the receptors on the pituitary cells effectively burn out.
They get overwhelmed.
They do. The cell says this is too much signal, and it pulls the receptors off its surface. It's a process called downregulation and internalization. They retreat into the cell, and they stop listening.
So a continuous signal is actually interpreted as noise by the system, and it shuts itself down.
Precisely. We actually use this clinically. If we need to medically shut down a patient's reproductive system, say for treating prostate cancer or severe endometriosis, we give them a long-acting, continuous GnRH agonist. It overloads the system and paradoxically turns it off.
So to turn it on, you need a pulse. To turn it off, you give a constant flood. That's incredible.
It is. So natural puberty relies on this rhythmic pulse. The GnRH pulses travel down that special portal blood system to the pituitary, which then responds by releasing FSH and LH follicle-stimulating and luteinizing hormone in corresponding pulses.
And those hormones travel through the bloodstream to the ovary.
And the ovary wakes up.
And we start to see the clinical progression. The L'Arche, which is breast development, pubarche, hair growth, and then finally menarche, the first period. But I think it's important to note that the first period doesn't necessarily mean the system is fully calibrated and online, does it?
No, not at all. That's a very common misconception. The first few cycles, sometimes for the first year or even 2, are often anovulatory.
Meaning you get the bleeding, but no egg is actually being released.
Correct. The estrogen from the early follicle development is enough to build up the uterine lining. Eventually, that lining becomes unstable and sheds, and it looks like a period. But that incredibly complex positive feedback loop, the one that's required to actually launch an egg that hasn't matured yet.
It's like the orchestra's warming up. The instruments are all playing, but they aren't quite in sync yet.
The drummer is playing a steady beat, but they haven't learned the big solo yet. That's a good analogy.
There's one other factor I want to talk about regarding the start of puberty, and that's metabolism. Both texts mention the hormone leptin. This feels like the body's way of doing a resource check, asking, can we actually afford this?
That's the core of the critical body weight hypothesis. Reproduction is unbelievably metabolically expensive. A single pregnancy consumes roughly 80,000 extra calories. Lactation consumes even more. From a purely evolutionary standpoint, it is a terrible idea to start a pregnancy if you are starving.
So the body needs a way to know if there's enough fuel in the tank.
And leptin is the fuel gauge. Leptin is a hormone that's produced by adipose tissue, by your fat cells. The more fat stores you have, the more leptin is circulating in your blood.
So it's a signal of energy sufficiency.
Yes. The hypothalamus reads that leptin level. If leptin is too low, the brain makes an executive decision. There are no energy reserves. We are in a deficit. And it keeps that GnRH pulse generator inhibited. It won't release the brake.
Which explains so much clinically. It explains why we see delayed puberty or amenorrhea, the loss of periods in high-level athletes, gymnasts, ballet dancers, or individuals with eating disorders like anorexia.
Absolutely. The body is making a very rational, very primal economic decision. It's saying energy intake is low, energy expenditure is high, we cannot afford to run the reproductive factory right now. Shut it down to save the organism.
Okay, so let's assume the system is a go, the pulses are firing, the leptin levels are high enough. We enter the master cycle, section 3, the engine. I really want to spend some time here because the dynamics happening inside the ovary are just brutal.
It is a process of ruthless selection. We often think of nature as this nurturing force, but ovarian physiology is a story of constant, massive attrition.
Let's just look at the raw numbers because they're staggering.
Okay. A female fetus at around 20 weeks of gestation has about 7 million primitive follicles, or oocytes. 7 million. That is the absolute maximum she will ever have.
The peak.
The peak. By the time that baby is born, that number has already crashed to about 2 million. By the time she reaches puberty, it's down to less than 300,000.
Wait, so before the system even turns on for the first time, more than 95% of the initial inventory is just gone.
Gone. Through a process called apresia, which is just programmed cell death, and it gets even more extreme. Over a woman's entire reproductive life, she will only actually ovulate about 400 to 500 of those eggs.
So the rest, the other 299,500.
They're also lost to atresia. It is a system designed for constant pruning and rejection.
So let's trace the journey of one of those lucky 400. We're at day 1 of the cycle. What's happening?
Day 1 is defined as the first day of menstrual bleeding. At this point, the hormones from the previous cycle, estrogen and progesterone, have crashed. They're at rock bottom. The pituitary senses this low hum and responds by slightly increasing its secretion of FSH.
Follicle-stimulating hormone. It does what it says.
It does. This little rise in FSH acts as a recruitment signal. It goes to the ovary and recruits a cohort of small antral follicles, maybe 10, maybe 20 of them. Think of them as runners being called to the starting line of a race.
They all have the potential to be the egg for that month.
They all do, but they are now in a race against time, and more importantly, in a race against each other.
And this brings us to the really elegant cellular mechanics of how these follicles grow. Ganong has a fantastic section on the two-cell, two-gonadotropin theory. It sounds technical, but it explains so much about why the system is regulated the way it is.
It's a critical concept to grasp. So a follicle is not just an egg, it's the egg, the oocyte in the middle, and it's surrounded by layers of support cells. For our purposes, there are 2 key layers. The inner layer is the granulosa cells. The outer layer is the theca cells.
And they have different jobs, but they have to work together.
They have completely different but totally complementary jobs. It's like a factory assembly line. The theca cells on the outside have receptors for LH. When LH from the pituitary binds to them, they take up cholesterol and convert it into androgens.
Androgens like testosterone.
Specifically, androstenedione and testosterone. So the first step in making the primary female hormone is actually making male hormones.
The ovary is an androgen factory first.
It is. But the theca cells are stuck at that point. They lack a key enzyme called aromatase, so they can't take that final step to turn the testosterone into estrogen. They can only make the raw material.
They have to pass it off to their neighbors.
They do. The androgens diffuse across a basement membrane into the inner layer to the granulosa cells. The granulosa cells have the FSH receptors. When FSH from the pituitary binds to them, it powerfully activates that enzyme, aromatase.
And aromatase is what does the conversion.
Aromatase grabs that testosterone from the theca cells and converts it into estradiol, which is the most potent form of estrogen.
So let me see if I have this straight. Theca cells use LH to make androgen fuel. Granulosa cells use FSH to light that fuel on fire and turn it into estrogen.
That's a great way to put it.
But why split the job? Why not just have one cell that does the whole thing?
It's all about control. By requiring two different hormones, LH and FSH, acting on two different cells to create the single final product, the body builds in a powerful control mechanism. It prevents runaway estrogen production. It forces the system to be coordinated and responsive to the brain.
Okay, so this whole cohort of 10 to 20 follicles, they're all running this two-cell factory. They're all pumping out estrogen. But we know that in humans, typically only one egg ovulates per month. So how is the winner chosen?
This is the selection phase. It's Darwinian. As all these follicles grow, they pump out more and more estrogen into the blood. As that estrogen level rises, it feeds back to the brain and the pituitary and tells them to suppress FSH secretion.
Standard negative feedback.
Standard negative feedback. So FSH levels, which were already low, start to drop even further. Now imagine you're one of those 20 follicles. Your survival depends on FSH. But the total supply of FSH is dwindling because you and your siblings are producing the very estrogen that's shutting off the tap.
It's a Hunger Games scenario. They're inadvertently poisoning their own well.
That's exactly what it is. And the follicle that survives is the one that is biggest, most mature, and has a secret weapon.
What's the weapon?
The dominant follicle, as it's called, has developed more FSH receptors than its competitors. So it's much better at scavenging the little bit of FSH that's left in the system. It can survive on the scraps.
It's more efficient.
More efficient. And this is the key detail that Guyton highlights. It also starts to acquire LH receptors on its granulosa cells.
So it changes its diet. It learns to use a different food source.
Exactly. It learns to respond to LH as a survival and growth signal, not just FSH. The other, smaller follicles can't do this. So as FSH levels plummet, they starve and undergo atresia. They die off. The dominant follicle is the only one left standing.
That is just incredibly elegant and incredibly ruthless. The winner effectively kills off the losers by cutting off their shared food supply.
It does. And now this single dominant follicle is a massive, unchecked estrogen factory. The estrogen levels in the blood, which were rising slowly, now start to skyrocket.
Which brings us to probably the most dramatic plot twist in all of physiology. Section 4, the control room. The great feedback flip.
This is the concept that I think confuses medical students the most because it breaks the rules we've taught them everywhere else.
Right. For the first 12 or 13 days of the cycle, estrogen is an inhibitor. High estrogen tells the pituitary, calm down, make less FSH and LH. It's negative feedback.
But for a crucial 24 to 48-hour window before ovulation, that rule completely reverses.
How? Why does the pituitary suddenly decide, you know what, actually, I love estrogen now?
It's known as the threshold hypothesis. It's not just about how high the estrogen level gets, it's also about the duration it stays high.
Okay, so it's two-part signal.
Yes. The estrogen level must exceed a certain critical threshold, usually over 200 picograms per milliliter, and it has to stay above that threshold for at least 36 to 48 hours continuously.
So the brain is basically giving the follicle a stress test. It's saying, are you really a mature, dominant follicle? Prove it. Show me you can sustain this incredibly high output.
Exactly. If the follicle can hold that high note for long enough, the hypothalamus and pituitary interpret it as the ultimate readiness signal. The negative feedback switches to powerful positive feedback.
Now, what does that look like?
The pituitary dramatically increases its sensitivity to the GnRH pulses coming from the hypothalamus and just dumps its entire stored reserve of LH into the bloodstream.
The LH surge.
The LH surge. It's a massive, sharp spike. Levels can increase tenfold or more in just a few hours. And this surge is the non-negotiable trigger for ovulation. No surge, no ovulation.
Physically, what is the surge doing to that dominant follicle?
Yeah.
It's been growing peacefully, and now it gets hit with this tidal wave of LH.
It turns the follicle from a growing balloon into a bursting grenade. The LH surge does a few things. First, it causes the granulosa cells to start secreting proteolytic enzymes, things like collagenase.
Enzymes that digest protein.
Exactly. They start digesting the wall of the follicle itself, the collagen matrix. It weakens the casing from the inside out.
It's dissolving its own container.
It is. At the same time, the surge causes the release of prostaglandins, which cause vasodilation and swelling in the follicle wall. So the pressure inside is rising while the wall itself is getting weaker and weaker
Until
it ruptures. A small nipple-like protrusion called the stigma forms on the surface. It bursts, and the rush of follicular fluid carries the egg out into the peritoneal cavity, where it can hopefully be picked up by the fallopian tube.
So ovulation is not a gentle, passive leak. It's an explosive, inflammatory-like event.
It is remarkably similar to an inflammatory reaction. In fact, if you give a woman high doses of anti-inflammatory drugs that block prostaglandins, like ibuprofen, you can actually inhibit the final rupture step, even if all the hormones are perfect.
That's a fascinating clinical pearl. Okay, so the egg is gone. It's on its journey. But the cycle isn't over. We enter the second half, the luteal phase. What happens to the empty shell of the follicle that's left behind in the ovary?
It transforms. This is another miracle of the ovary. The cells don't just die off yet. Under the profound influence of that same LH surge, the theca and granulosa cells luteinize.
They change their function.
They completely change. They start accumulating lipids and a yellow pigment called lutein.
Which is where we get the name corpus luteum, the yellow body.
Precisely. And they switch their primary product. They still make some estrogen, but now they become a factory for pumping out massive quantities of progesterone.
The progestation hormone.
The progestation hormone. If estrogen was the get-ready hormone that built the house, progesterone is the stay-ready hormone. It makes the house hospitable. It calms the uterine muscle. It slightly suppresses the local immune system to allow for implantation. And it creates a nutrient-rich, receptive environment.
But the corpus luteum has a fatal flaw, doesn't it? It's on a timer.
It is programmed to self-destruct. It has an intrinsic lifespan of about 14 days. If it doesn't receive a new external rescue signal by that time, it undergoes luteolysis. It dies.
And it shrivels up into a little scar.
A scar called the corpus albicans, the white body.
And when it dies, the progesterone that it was producing crashes. And that crash is the signal that resets the entire system for the next month.
That's the reset button. But before we get to that, let's look at the other player in this duet.
Yeah.
We can't forget the uterus. Section 5. The uterine or endometrial cycle.
Right, we've been laser focused on the ovary, which is the director. But the uterus is the stage. It's the one doing the heavy lifting in terms of visible tissue change.
The uterus is the ultimate responder. Its cycle perfectly mirrors the ovarian cycle. So when the ovary is in its follicular phase, and it's secreting mainly estrogen.
The uterus is in what's called the proliferative phase.
Proliferative just means growth. Remember, the uterine lining, the endometrium, was just shed during the last period. So now, under the influence of rising estrogen, the basal layer starts to regrow. The glands lengthen, the stroma thickens, new spiral blood vessels form. It's a period of rapid reconstruction.
Rebuilding the house after the last tenants left.
Exactly. And a key job of estrogen here is to induce the formation of progesterone receptors on those endometrial cells. It's priming the tissue for the next phase.
So then ovulation happens.
Yeah.
The ovary switches to making progesterone. It enters the luteal phase. The uterus then switches to the secretory phase.
Right, and this phase is about quality, not just quantity. The rapid growth stops. Under progesterone's influence, the glands become tortuous and coiled, like little corkscrews, and they start actively secreting glycogen and lipids into the uterine cavity.
Guyton calls this uterine milk.
It's stocking the pantry for a potential embryo. It's making the lining not just thick, but nutritious and receptive.
So progesterone has another critical job, right?
Yeah.
Calming the muscle.
Yes, it quiets the myometrium, the uterine muscle, to prevent contractions that might expel a newly implanting embryo. And it also has a systemic effect. Progesterone is thermogenic. It raises a woman's basal body temperature by about 0.5 degree Fahrenheit, which is a clinical sign that ovulation has occurred.
The timing of all this is critical. There's a window of implantation.
There is. The uterus is only receptive to an embryo for a very short, specific time, usually from about day 20 to day 24 of the cycle. If an embryo arrives too early or too late, the molecular door is locked. It can't stick.
Timing is everything. So let's follow the default path. No embryo arrives. That 14-day clock on the corpus luteum runs out. It dies. Progesterone and estrogen levels plummet. What is the actual mechanism of menstruation?
It's fundamentally a vascular event. Progesterone was the hormone responsible for keeping those special spiral arteries in the endometrium relaxed and open. When progesterone is withdrawn, those arteries go into intense spasm.
They constrict.
They constrict, cutting off blood flow to the upper layers of the endometrium. Then they might relax, then they spasm again. This causes the tissue to become ischemic. It loses its oxygen supply.
It's like inducing a tiny heart attack in the uterine lining.
That's an excellent analogy. The tissue becomes necrotic and dies. Lysosomes within the cells rupture, releasing enzymes that digest the dying tissue, and this dying tissue itself releases prostaglandins.
Prostaglandins again.
Which cause the uterine muscle, the myometrium, to contract. Those are cramps. Their job is to help expel all the blood and necrotic debris.
And that shedding brings us right back to day one. The cycle resets. Clean slate.
But the story doesn't always end in a reset. And this brings us to Section 6, adaptability. What happens if the system actually works? Pregnancy.
The rescue mission.
Right. So if an egg is fertilized in the fallopian tube, it begins to divide. It travels to the uterus, and around day 6 or 7 after ovulation, the blastocyst, as it's now called, implants into that receptive endometrium.
And it has to send a signal. It has to announce its presence.
It has to, and it has to do it quickly before the corpus luteum dies. The outer cells of that new embryo, the syncytiotrophoblasts, start secreting a unique hormone that the mother's body doesn't make.
HCG, human chorionic gonadotropin.
The pregnancy test hormone. Now, HCG is a fascinating molecule. Structurally, it is almost identical to LH. It's like LH's molecular twin. So it gets into the mother's bloodstream, it travels to the ovary, and it binds to the LH receptors on the corpus luteum.
So it's basically impersonating LH to trick the corpus luteum.
It sends it a powerful signal. It says, don't die, we need you, a pregnancy has started. The corpus luteum is rescued from its programmed death. It stays alive and continues to pump out that essential progesterone.
And because progesterone stays high, the uterine lining is maintained, the spiral arteries don't spasm, the period is missed.
And the pregnancy is sustained. That handover from the mother's brain driving the cycle to the embryo itself driving the cycle via HCG is the single most critical moment of early pregnancy.
And that corpus luteum keeps the pregnancy going for how long?
For the first 8 to 12 weeks. Eventually, the placenta grows large enough to take over as the main factory for progesterone and estrogen, but for that entire first trimester, the corpus luteum of pregnancy is the absolute life support system.
So that's one interruption of the cycle. Let's talk about the final one, the end of the line, menopause. It's often described simply as running out of eggs, but chemically it's quite a dramatic shift.
It is. As a woman approaches her late 40s and 50s, the pool of remaining follicles is exhausted. And as that pool dwindles, 2 things start to happen. First, you lose a hormone called inhibin.
We haven't talked much about inhibin.
Inhibin is produced by those granulosa cells in the growing cohort of follicles, and its main job is to provide specific negative feedback on FSH. It keeps FSH in check during the follicular phase.
So before you even lose estrogen, you start to lose inhibin.
Yes. This is the period of perimenopause. Inhibin levels drop because there are fewer follicles being recruited each month. As inhibin drops, its brake on the pituitary is released. So FSH starts to creep up. The brain is starting to have to shout louder to get the aging ovaries to respond.
The accelerator is being pressed harder just to maintain speed.
Exactly. But eventually you reach a point where there are virtually no follicles left to recruit at all. No follicles means no granulosa cells, which means no more estrogen production.
And without the powerful negative feedback from estrogen.
The pituitary essentially goes unregulated. It screams at the silent ovaries. It pours out massive, massive amounts of FSH and LH, trying desperately to get a response that can never come.
So the hormonal signature of menopause is actually super high, FSH and LH.
Sky high. If you draw blood from a postmenopausal woman, her FSH level can be 10 to 20 times higher than that of a young woman. It's the diagnostic hallmark. The brain is shouting into the void.
And the silence from the ovaries has consequences throughout the body.
Absolutely. Estrogen isn't just for making babies, it's crucial for maintaining bone density, so its loss leads to osteoporosis. It helps maintain vascular health, so cardiovascular risk goes up. It maintains the health of vaginal and urinary tissues. The loss of this cycle affects the entire organism.
Wow. So let's try and bring this all together. We've gone from the SRY gene on the Y chromosome acting as the master switch to the hypothalamic pulse generator waking the system up, through the brutal Hunger Games of follicular selection, and finally to the chemical rescue of a pregnancy.
It is a system that is completely defined by its dynamics, by its rhythm, by its cycles.
For me, the single biggest takeaway, the theme that runs through this whole story, is this concept of rejection by default. The body seems designed at every single checkpoint to say no.
That's a really profound insight. The default state for an oocyte is atresia death. The default state for the uterine lining is menstruation shedding. The system is fundamentally biased towards resetting and starting over unless the conditions are absolutely perfect.
The follicle has to be perfect to survive that FSH drop.
The hormonal surge has to be timed perfectly to trigger ovulation.
And the embryo has to shout its presence with HCG to stop the whole thing from being washed away.
It's a need-to-know system. The uterus has no idea if fertilization happened. The brain has no idea. They are all waiting for that one positive chemical signal from the embryo. Without that signal, the system ruthlessly resets.
It's a system of immense potential, but it's governed by these incredibly strict chemical communication protocols.
It ensures that the massive metabolic investment of pregnancy is only made when success is at least plausible. It prioritizes quality control over raw efficiency.
If you are listening to this and you want one thing to do to really cement this knowledge, I have a homework assignment for you. Open your gynecologist textbook to the chapter on the female reproductive cycle and find what is probably figure 82-4.
Ah, the Rosetta Stone.
It is. It's that famous graph with the 4 hormone lines plotted over the 28-day cycle. FSH, LH, estrogen, and progesterone. Don't just look at it. Take your finger and trace it. Narrate the story we just told out loud.
Point to the graph and say, here is the FSH recruiting the follicles. Here is the estrogen rising. Here is where it hits the threshold and flips to positive feedback. There's the LH surge. And here is the corpus luteum dying and taking progesterone down with it.
If you can narrate that graph from start to finish, you truly understand the female reproductive cycle.
It all comes together in that one figure.
Thanks for diving deep with us today. Keep asking questions, and remember, physiology isn't just a list of parts. It's a living, breathing logic.
Stay curious.